OliX's OLX 501A targets ALK7 in adipocytes. Rather than simply reducing body weight through the loss of both fat and muscle, OLX 501A is designed to improve the quality of weight loss by reducing fat while preserving muscle mass, and that potential is starting to be confirmed by data. In mouse models, siRNA was effectively delivered to adipose tissue following subcutaneous administration, while effects on major organs such as the liver and kidneys were limited. In addition, when administered at 3 mg per kilogram, more than 80% suppression of gene expression was observed, and this effect lasted for over 2 months. In obese mouse models, administration of OLX 501A alone resulted in a 10.7% reduction in body weight compared with the control group after only 28 days of treatment. Furthermore, after 28 days of treatment, body fat was reduced by 32.6% compared with the control group. The body composition data also demonstrated the potential to selectively reduce fat while preserving muscle mass. Moreover, in obese mouse models, when OLX 501A was administered in combination with a low dose of Zepbound, the efficacy of body fat reduction was maintained even at 1/10 the dose compared to Zepbound high-dose monotherapy. Additionally, muscle loss was significantly alleviated, making simultaneous reduction of body fat and preservation of muscle mass possible. We have confirmed the potential for quality improvement in weight loss. OliX is currently optimizing OLX 501A to achieve its goals of gene expression inhibition, body fat reduction, and improved weight quality. And now we want to take it one step further here. Hello, I am June Park, the head of Research Division. In previous session, our CEO has overviewed our programs. The content I will explain is more detailed and based on data about ALK7 program and dual targeting platform. As I mentioned earlier, we have developed a new adipose targeting platform. The schematic shown here was generated using AI to help illustrate the overall concept. While it is not intended to represent the exact molecular structure, it provides a good overview of the platform design. At its core, the siRNA backbone is highly stable as it has been engineered to ensure long-term stability. What differentiates our adipose targeting platform from conventional lipid-conjugated siRNA platforms is its dual lipid architecture. Delivery efficiency is determined by the specific chemistry of each lipid component. Because minimizing liver uptake is critical, we design our lipid conjugates with chemistries that reduce hepatic accumulation. We invested considerable effort into optimizing the design. As a result, compared with the previous generation, the current version demonstrates excellent target knockdown, with robust activity maintained for more than 2 months, even at a low dose in mice. Importantly, the platform is also designed to minimize delivery to non-target tissues, particularly the liver and kidneys. After establishing our adipose targeting platform, the first program we initiated was ALK7, which I'll briefly introduce today. In obesity, the expression of multiple ALK7 ligands is upregulated. These include inhibin E, GDF3, and other members of the activin family. Binding of these ligands activates ALK7 signaling in adipocytes. Once activated, the ALK7 SMAD pathway suppresses lipolysis, preventing stored fat from being mobilized and used as an energy source. As a result, fat continues to accumulate within adipose tissue. Our approach is to silence ALK7 using siRNA. By inhibiting ALK7, we remove the brake on lipolysis, allowing stored fat to be mobilized, broken down, and used as an energy source. In other words, our mechanism of action reactivates the fat-burning capacity of adipose tissue. GWAS analyses of individuals carrying ALK7 loss-of-function variants have shown multiple metabolic benefits, including reduced abdominal fat, smaller waist circumference, and a lower risk of type 2 diabetes. Importantly, no specific safety concerns have been associated with these variants. OliX is a highly validated target, and recent clinical data from Arrowhead have provided encouraging proof of concept in humans. To briefly explain the difference from the Inhibin E approach, Inhibin E is only one of several ligands that activate OliX. Therefore, blocking a single ligand is expected to have a more limited effect than directly inhibiting the receptor. By targeting OliX itself, we can block signaling from multiple ligands simultaneously, potentially resulting in a broader and more robust therapeutic effect. Before moving to the monkey data, which will be the main focus of today's presentation, I'd like to briefly review our earlier mouse studies. These studies are still ongoing. As you can see, the duration of activity is remarkably long. When we first established this platform and compared it with GalAC-based platforms, one of the things that impressed us most was how durable the pharmacological activity was. This prolonged activity may be related to the unique characteristics of adipocytes. If the effect lasts for about 3 months in mice, we would expect an even longer duration in monkeys and potentially in humans. The treatment achieved approximately 10% body weight reduction. However, this program is not designed to maximize overall weight loss. Rather, it is specifically optimized to reduce body fat. Accordingly, most of the weight loss is attributable to fat mass reduction. In our mouse models, we have demonstrated reductions in body fat of up to 45%. Based on these encouraging results, we conducted a head-to-head study in an obese non-human primate model, comparing our development candidate with a competitor compound that is currently in clinical development. As this is a formal presentation, I hope you understand that we are not able to disclose the identity of the competitor. For this study, obese monkeys were selected based on predefined body weight and body fat criteria. Both compounds were administered at 3 mg per kilogram, with 2 doses given 1 month apart. Target knockdown was evaluated on days 28, 70, and 119 according to the study protocol. With regard to efficacy assessment, target knockdown in visceral adipose tissue would ideally be the most relevant pharmacodynamic endpoint. However, repeated visceral fat biopsies cannot be performed safely in non-human primates. Therefore, target knockdown was assessed in subcutaneous adipose tissue instead. Because subcutaneous target knockdown alone does not fully predict efficacy in visceral fat, we included several additional efficacy endpoints in the study. Most importantly, visceral fat volume was measured by MRI on day 49, which we consider the primary efficacy endpoint. At the end of the study, DEXA was performed to evaluate overall body composition, including both fat mass and lean mass. The data I will present today are interim results, including target knockdown on days 28 and 70, together with the MRI-based visceral fat analysis from day 49. The data I'm presenting today are from this study. Compared with the competitor, our candidate achieved comparable or slightly superior OliX gene knockdown. The knockdown was rapid, reaching approximately 90% and remaining at that level through day 71. We expect the effect to be even more durable beyond this time point. More importantly, the reduction in visceral fat was highly impressive, approximately 3-fold greater than that observed with the competitor. Taken together, these findings suggest that our candidate is likely achieving stronger OliX inhibition in visceral adipose tissue than the competitor, even though target knockdown was measured in subcutaneous fat. In addition to target knockdown, we evaluated ADRB3, a pharmacodynamic biomarker that is closely linked to OliX inhibition. When OliX is suppressed, adipocytes become more responsive to norepinephrine, which promotes lipolysis. Because ADRB3 is a key receptor that mediates this response, increased ADRB3 expression reflects enhanced sensitivity to norepinephrine and a stronger fat-burning signal. For this reason, ADRB3 serves as a useful pharmacodynamic biomarker of OliX inhibition, providing functional evidence that the target is being effectively engaged. When we measured ADRB3 expression on days 28 and 70, Our compound induced approximately a twofold greater increase than the competitor at day 28, and this advantage appeared to be maintained through day 70. Taken together, the robust ALK7 knockdown in subcutaneous fat, the marked reduction in visceral fat, and the favorable changes in this key pharmacodynamic biomarker, we believe these three independent lines of evidence consistently support the best-in-class potential of our compound. Looking at the competitive landscape, there are currently 3 companies developing ALK7 targeting therapies: Arrowhead, Alnylam, and Seron Bio. Arrowhead is the most advanced with its program in phase 1 and 2 clinical development and has recently reported encouraging early clinical data demonstrating reductions in liver fat and liver enzymes. Meanwhile, the programs from Alnylam and Seron Bio remain in the early stages of clinical development. As you know, both Alnylam and Arrowhead are developing their own proprietary ALK7 programs and are therefore unlikely to pursue partnering opportunities in this area. However, Seron Bio recently entered into a licensing agreement with GSK for its ALK7 program. In addition, although the specific targets covered in Sanogene Bio's partnership with Genentech have not been disclosed, we believe ALK7 is likely included. To our knowledge, ours is the only competitive preclinical ALK7 asset that remains available for partnering. As such, we believe there is a meaningful opportunity for out-licensing, either during the current preclinical stage or in early clinical development. Everything is progressing according to plan. Based on our current timeline, we expect to initiate clinical trials in the first half of next year. In addition to ALK7, we are actively expanding our obesity pipeline with additional adipose-related targets. Let me briefly introduce one example, a novel target involved in adipocyte metabolism. Although these data are still preliminary, we have observed greater body weight and fat mass reduction than with ALK7. In addition, the compound achieved highly efficient target knockdown of over 95%. We plan to disclose the identity of this target at the appropriate time. The key message of this slide is that our adipose targeting platform has proven to be both robust and highly reproducible. Regardless of the target gene, the platform consistently delivers highly efficient target knockdown, demonstrating that its performance is driven by the strength of the delivery technology rather than by any single target. Given the strong therapeutic potential of ALK7 in obesity, our strategy is to build a dual-targeting adipose platform centered on ALK7. Although I won't be presenting those data today, we are actively developing dual-targeting siRNA programs that combine ALK7 with complementary targets involved in adipose biology and lipid metabolism to further enhance efficacy. At the same time, we are rapidly evaluating multiple obesity targets in parallel to efficiently expand and diversify our pipeline. Rather than starting with large-scale screening, we first identify a small number of promising hits and rapidly evaluate them in animal proof-of-concept studies. When a target demonstrates a compelling efficacy profile, we then concentrate our optimization and screening efforts around that program to accelerate lead identification and pipeline expansion. Using this approach, we are able to advance multiple obesity programs in parallel while maintaining both speed and efficiency. Our overall strategy is to use ALK7 as the foundation of our obesity pipeline and combine it with other promising novel targets through a dual-targeting approach. We believe obesity is a highly heterogeneous disease, and many patients require metabolic interventions beyond GLP-1-based therapies alone. By targeting complementary pathways with siRNA, we believe we can address a broader range of patient populations and create differentiated treatment options. Next, I'd like to briefly introduce our OASIS-D platform. OASIS-D is our dual-targeting platform designed to simultaneously silence 2 targets with a single siRNA molecule. I'll just give you a brief overview and a quick update on our recent progress. For ocular indications, we recently completed development of the OASIS-D platform through continuous optimization of our lipid conjugate chemistry. Using this platform, we demonstrated that a single siRNA molecule can simultaneously silence 2 targets not only in the liver but also in ocular tissue. More importantly, the dual-targeting siRNA achieved superior efficacy compared with an equivalent dose mixture of the corresponding single-target siRNAs. As many of you know, the key advantage of the OASIS-D platform is that a single molecule can simultaneously modulate two therapeutic targets. Because both targets are inhibited by the same molecule within the same cells, the platform has the potential to provide more consistent PK and PD than conventional combination therapies. It also offers meaningful advantages from a CMC perspective by simplifying manufacturing and development. These advantages are particularly valuable for ocular diseases where intravitreal injections are invasive and place a significant burden on patients. By enabling simultaneous modulation of two targets with a single intravitreal injection, we believe OASIS-D has the potential to improve patient convenience while delivering differentiated therapeutic efficacy and a meaningful competitive advantage. Let me now provide a brief update on one of our GalNAc-based dual-targeting programs for liver diseases. The data shown on the left are from our cardiovascular program, which we presented earlier this year. In that work, we identified a pair of targets that produced synergistic reductions in cholesterol, and particularly triglycerides. Based on those findings, we designed a dual-target siRNA and are currently selecting the development candidate. Next quarter, we plan to initiate studies in a dyslipidemic non-human primate model to further evaluate the efficacy and translational potential of this program. Our other major GalNAc-based dual-targeting program focuses on obesity and metabolic disease. For this program, we have already selected the target combination, completed molecular design, and initiated non-human primate studies. Unfortunately, due to our contractual obligations with our partner, we are not able to disclose the data at this time. We appreciate your understanding. The monkey studies are progressing well, and by next quarter, we expect to generate a comprehensive large animal data package, including PK, PD, and exploratory toxicology to support advancement into the next stage of development. Our current ocular dual-targeting strategy is built around OliX 301A as the backbone, combined with either a geographic atrophy target or a wet AMD target to maximize therapeutic efficacy in both indications. At the same time, we have continued to advance the platform itself. OliX 301A has been upgraded to our next-generation chemistry, and more recently we identified an additional novel target for geographic atrophy, further strengthening our ocular pipeline. We first evaluated each molecule individually in a geographic atrophy model. In the fundus images on the left, the bright areas represent regions of retinal cell damage. As you can see, both OliX 301A Gen 2 and the newly identified GA target demonstrated strong therapeutic activity, showing clear improvements in both fundus imaging and ERG-based assessments of retinal function. Based on these encouraging results, we are now developing a single dual-target siRNA that combines these two targets, with the goal of achieving even greater therapeutic efficacy than either approach alone. Our dual-target molecule consists of two components. One is the second-generation version of OliX 301A, which incorporates our further optimized chemistry, and the other is a newly identified target for geographic atrophy. Recently, we have focused much of our development effort on GA. As many of you know, OliX 301A has already demonstrated robust efficacy in our GA models. In addition, the novel GA target, shown in blue on this slide, has also shown strong therapeutic activity. On the left are representative fundus images from the GA model. The bright areas indicate regions of retinal damage, while the darker areas represent relatively healthy retinal tissue. As shown in both the fundus images and the ERG-based assessments of retinal function, we confirm strong efficacy with both the siRNA targeting the new GA target and our second-generation OliX 301A. These results provided the rationale for combining the two into a single dual-target molecule to further enhance therapeutic efficacy. Based on these findings, we designed a dual-target siRNA combining these two targets and recently completed our first proof-of-concept study. The treatment groups included second-generation OliX 301A, target X alone, and the dual target siRNA. Therapeutic efficacy was evaluated in the GA model using ERG as a measure of retinal function. To rigorously assess the benefit of dual targeting, the dual target siRNA was administered at half the dose of each individual siRNA, ensuring that the total siRNA dose was equivalent across all treatment groups. As you can see, even at half the dose per target, the dual target siRNA provided the greatest preservation of retinal function. These findings demonstrate that our dual targeting platform is not only mechanistically sound, but also capable of delivering superior therapeutic efficacy compared with single target approaches. Building on these encouraging results, we plan to further expand our ocular pipeline and generate a comprehensive development package, including safety and large animal studies to support future partnering opportunities and clinical development. Finally, I'd like to introduce our dual organ targeting platform. We chose to begin with the liver and adipose tissue because we believe they are the two most important organs in regulating metabolic disease. Traditionally, adipose tissue was viewed simply as a site for fat storage, while the liver was regarded as the primary metabolic organ. However, our understanding has advanced significantly in recent years. It is now well established that the liver and adipose tissue communicate extensively through hormones, metabolites, and other signaling molecules, forming an integrated metabolic network that plays a central role in obesity and metabolic disease. In fact, metabolic diseases such as obesity and MASH are rarely disorders of a single organ. Instead, they involve dysfunction in both the liver and adipose tissue. While therapies that target either organ alone can certainly provide meaningful benefit, we believe that simultaneously modulating both organs has the potential to deliver greater therapeutic efficacy for metabolic diseases such as obesity and MASH. As we first demonstrated with OliX 501A, our approach is to conjugate GalNAc on one end to target the liver and an adipose-targeting lipid ligand on the other end. The two components are connected by a linker that is designed to be cleaved in vivo. To the best of our knowledge, this dual organ targeting platform is unique to OliX. Even if similar approaches emerge in the future, we believe we are among the pioneers and leaders in this area. We have already filed patent applications covering this platform to protect its core technology. Here, I'd like to share some of the early data we have recently generated. In this mouse study, we administered our dual organ construct and confirmed efficient target knockdown in both the liver and adipose tissue. The targets used in this experiment are well-established model targets for demonstrating liver and adipose delivery. On the left, you can see the liver data, where target knockdown was highly efficient and comparable to that achieved with a conventional GalNAc conjugated single-target siRNA. Importantly, we observed similarly robust target knockdown in adipose tissue. Although this platform is still in the early stages of development, these results demonstrate that we have successfully established a highly effective dual organ targeting platform. Building on these encouraging data, we plan to expand this technology into multiple metabolic disease programs. Finally, I'd like to conclude by sharing our broader platform vision. While our current focus is on dual targeting of the liver and adipose tissue, we believe the same platform can be readily extended to other organ combinations, such as liver-kidney and adipose muscle. In parallel, we are also exploring triple organ targeting in collaboration with our chemistry team in San Diego. Although it is still at an early stage and we do not yet have data to disclose, we believe this represents an exciting next step for the platform. Importantly, triple targeting does not necessarily mean targeting 3 different organs. It could, for example, involve 2 liver targets together with 1 adipose target. Looking ahead, we will continue to advance our differentiated delivery platforms and strengthen our intellectual property. We believe these technologies will not only expand our internal pipeline, but also create attractive opportunities for platform-based collaborations and strategic partnerships. With that, I would like to conclude my presentation. Thank you very much for your attention.